this is my membrane concept map. Okay, so we'll just get started. I started with the plasma membrane in the middle, and I branched out from there. Here we have a peripheral membrane protein, and those are proteins that only temporarily adhere to the plasma membrane. And then we go to selective permeability, and that is a feature and function of the plasma membrane that is necessary to maintain homeostasis. Here, and then we'll go to sterols, which are they determine the shape of the plasma membrane. Vesicles here. They are small, membrane-enclosed spherical organelles in the cytoplasm of a eukaryotic cell. Then we'll go down to phospholipid bilayer, which it is the two-layered arrangement of phosphate and lipid molecules that form a cell membrane. And then we'll go ahead and travel down from phospholipid bilayer to hydrophilic and head and hydrophobic tail, and those make up the membrane lipid molecule. And hydrophilic is, of course, water-loving, and hydrophobic is water-fearing. And then from these we can get detergent molecules, and detergent molecules can manipulate hydrophobic and hydrophilic interactions among molecules. And then we also have amphipathic, I'm not sure if I pronounced that correctly, but here we have it going to both hydrophobic and hydrophilic, just like the detergent molecule. Anyway, amphipathic has both hydrophobic and hydrophilic regions. An example would be a phospholipid or detergent molecule. And also coming from the hydrophobic tail of phospholipid, I have a glycolipid, and it is a membrane lipid molecule with a short carbohydrate chain that is attached to a hydrophobic tail. Okay, and we'll go back up to the plasma membrane and go down to integral membrane protein, which is a type of protein that is permanently attached to the membrane. And then we can go down to simple diffusion, which simple diffusion is a substance, is when a substance can pass through a membrane without the aid of an intermediary, which would be the integral membrane protein. Also from integral membrane protein, we can go down to membrane proteins, which is a protein associated with the lipid bilayer, and they can either be integral or peripheral. I went ahead and grouped it with integral. From membrane proteins, we have all the different types of proteins. We have transporter membrane protein, which is a type of protein that actively transports materials across the plasma membrane. We have anchor membrane proteins, which are covalently bonded to a fatty acid and anchors them to either face of the cell membrane. Receptor membrane proteins, they are specialized in specialized integral membrane proteins that take part in communication between cell and outside. Protein attached membrane associated protein is a protein that is attached to or associated with a membrane. Monolayer associated membrane protein. We have membranes consist of two monolayers of protein molecules with a double layer of lipids between. And then we can go over to lipid-linked membrane-associated protein, which are proteins that are covalently linked to lipid molecules or hydrophobic molecules. Enzyme membrane proteins, which is when an enzyme is present or embedded in the membrane. I also attached proteoglycan to membrane proteins because it is a molecule consisting of one or more I cannot pronounce this word, glycosaminoglycan chains attached to a core protein, so I went ahead and put it with all the proteins. And then I also linked glycoprotein, which is a protein with one or more covalently linked oligosaccharide chains. Okay, we can come back up to plasma membrane. We have the gated ion channel, which is transmembrane ion channel that channel proteins that open to allow ions such as sodium, potassium, etc. to pass through the membrane. Yes. And then we have the membrane channel, which allows water and other inorganic ions to passively diffuse across the membrane bilayer. And then we can move from membrane channel to channel-mediated passive transport, which allows only one type of solute to pass through. And back up to the plasma membrane, we have active transport and passive transport. Active transport is the movement of ions or molecules across a cell membrane into a region of higher concentration, and passive transport is the movement of a small molecule or ion across membrane due to a difference in concentration or electrical charge. So from passive transport, we can go to electrochemical gradient, which is the driving force that causes an ion to move across a membrane. And then we can go from electrochemical gradient to ion channel, which is the protein that forms a water-filled channel across the lipid bilayer through specific inorganic ions and can diffuse down their electrochemical gradients. Back to passive transport, we can go to concentration gradient, which is the measurement of how the concentration of something changes from one place to another. So then we can go to osmosis, which is the net movement of water molecules across a semi-permeable membrane driven by a difference in concentration of solute on either side. And then we can go to our isotonic, hypertonic, and hypotonic solutions. And those basically, you either have like an isotonic, you have the same osmotic pressure on either side, and hypertonic, you have a higher concentration of solutes on the outside than on the inside, and then hypotonic, you have a lower osmotic pressure on the outside. And then I kind of went away from this main map, and I have unsaturated fatty acid and saturated fatty acid linked together, and they both have carbon chains. That's like, I guess, their main similarity. A saturated fatty acid has a carbon chain that contains no unsaturated links between carbon atoms, and therefore it cannot incorporate any more hydrogen atoms. And then we have unsaturated fatty acid, which is a fatty acid where the carbon chain can absorb additional hydrogen ions. Then up here, I put our two models. Of course, the sandwich model by Davison and Danieli was the first plasma membrane proposed in 1935, and it describes a phospholipid bilayer that lies between two layers of globular proteins. And then the one that we accept today, the fluid mosaic model by Singer and Nicholson, proposed in around the 70s, and it is a model where the cell membranes are composed of a double layer of lipids with associated proteins. And that's it. I'll zoom it up so that you can see more. And I'll just kind of move it across so you can get everything. So we have the plasma membrane, and then over here we have all of the concentrations. And down here we go to all the different proteins. And hydrophobic, hydrophilic, target molecules. And down here we have unsaturated fatty acid and saturated fatty acid. And then the two